Okay, good morning, everyone. Thank you so much for attending our webinar. We're going to go ahead and get started. So the webinar is going to run from 9 to noon. We will have two 10-minute breaks. So please be sure to pay attention to the talks because passing the poll questions and the quiz at the end are important for getting CEU credits. And know that the breaks will be coming. A little bit quick housekeeping. We're going to be recording this webinar for educational or promotional use. You will be muted during the webinar. Please communicate with us via chat for comments and questions. We will be tracking the questions for our speakers to answer at the end of their presentation. If you chat all panelists, that's only going to the speakers. And if you chat all panelists and attendees, that's going to go to everybody who's on this webinar. So it's up to you how you want to ask questions or make comments. And if you want to receive continuing education credits, you have to stay on the webinar throughout the entire duration. You cannot log on or off. So during the breaks, please don't log off. Just leave the screen up and step away and come back. You have to answer all the poll questions. You don't have to get the poll questions right, but you have to answer all the poll questions during the presentations. And then for CCA credits, you'll be scanning the QR code or replying to the post-webinar email. And for DPR credits, there is going to be a final exam that will be sent out in a post-webinar email, and you have to get a 70% or better to get your CEUs. The exam is based on the poll questions, so pay attention to the poll questions in order to ensure a good pass rate. Our agenda, I'm doing the welcome now. Our first presenter is Whitney Brim-DeForest. She'll be talking about weeds in rice research update. Then Luis Espino will be talking about rice pest management. We'll take a 10-minute break at 10:05, and then we'll go into an integrated pest management for processing tomatoes by veg crop advisor Amber Vinkesi-Val. I'll be talking about field crop pest management, and then we'll wrap up with regulatory updates by Scott Bowden from the Agricultural Commissioner's Office. We'll have 5 minutes at the end for a final wrap up in Q&A. And with that, we're going to go ahead and get started. I'm going to stop sharing my screen, and Whitney, go ahead and pull your slides up. Thanks. Thanks for attending.
Okay, good morning, everyone. Can everyone hear me okay?
Yep.
Okay. I'll go ahead and get started. So I'm going to be talking about weeds in rice, and this is a bit of a research update from what we've been doing in the past year. So the first thing I'll cover is our weed survey. So in 2019, right after harvest, we did a weed survey to kind of get a comprehensive idea of what we have in terms of weeds across the valley. We collected soil samples after harvest from across all of the rice growing regions. You can kind of see on the map here where we collected samples from. We got 10 randomly selected sites in most of the bigger counties. And then in some of the smaller counties, we got a few less sites just because the counties are a bit smaller in terms of rice growing area. So Yolo, Yuba County, for example, we got only 5 sites each. Placer and Sacramento, just 2 because they're very small. We sampled all of the major rice growing weeds. Whoops, sorry. Barnyard grass, bulrush, duck salad, our Echinocloa watergrass species, monocoria, red stem, small flower, sprangle-top, water hyssop, cattail, and rice. And this isn't completely done. I have a little bit, a little bit more data analysis to do. But basically, we grew the weeds out in the greenhouse for a few months to see what came up in the soil. The soil was again collected after harvest, and that was to ensure that we weren't, when we were doing the survey, that we were getting all of the possible weeds that could come up in rice. If we did it during the season, there would be a possibility that we would not see everything that was in the field because folks put out herbicides. So we did it in the fall, soil sampling to kind of make sure that we got a comprehensive overview of the weed species. And there weren't a whole lot of differences, I will say. Some of the key differences are that we saw monocoria really only in Butte County. Monocoria is one of the weed species that's on the noxious weeds list for the United States, for the entire US. And so it was important to be able to show where that was located on a map in comparison to the other rice counties. We did see a lot of rice come up, which is, I think, because we were sampling in the fall and there was a lot of rice on the ground. There was some weedy rice mixed in with the rice that we saw coming up. So that was good that we were able to show that as well in the survey. And I didn't differentiate between the rice and weedy rice yet. But basically, the idea with this is that we'll have a map that will be available up on the UC Rice website that will show sort of the distribution across the valley of our different weed species. Red stem and smallflower were present pretty much in every field that we sampled, as water hyssop was as well, and duck salad. Everything else varied a little bit by region. But hopefully this will be up on the UC Rice website within the next few months. So the two weeds that I'm really going to focus on today are watergrass and weedy rice. So I'll start with watergrass. So we've been dealing with an unknown watergrass species or biotype for the past few years. And to give everyone kind of a background in that, we had 2 fields that were identified in 2017. From that, Luis and I collected 10 populations for herbicide screening in 2018. We did some ID work in 2017 with our UC Davis herbarium, Ellen Dean there, and the results were inconclusive. Ellen sent those, I don't know, 15, 20 samples that we had two different Echinocloa experts, one in Texas and one in Canada, and the results were still inconclusive. So it has become apparent that we need to do some genetic work, but the genetics work to do this is pretty expensive. So we're starting with some herbicide screenings and phenotypic analysis, kind of like we did with the weedy rice. So looking at how the different biotypes or samples that we collect, how they look. So I'm going to focus on the herbicide screening here. So we collected 10 populations in 2018. 2 known susceptible populations were added as a comparison. These were samples that were collected from the Rice Experiment Station. We know that they're susceptible to our herbicides, or fairly susceptible. We screened them using our highest labeled field rate. It was conducted last spring in the greenhouse. The herbicides were applied at the 2-leaf stage, so it's very early application timing, and that's usually what we do for resistance screening. In the field, that would correspond to something between a day of seeding up to about 1.5 leaf stage of rice application, which is very early, obviously. So you'll see when I show the foliar herbicides that most of them work very well. The percent control we did with biomass and number of plants, and we did that at 14 days after application, those 2 evaluations, and then it was replicated 4 pots per biotype. Okay, so to get an idea of what it looked like for the susceptible types, you can see on the left here, this is percent control by biomass, and our herbicides down here on the bottom. So the first 4 or so are the granular herbicides that we apply at the beginning of the season. The other 3 are typically foliars that we apply usually at the 3 to 4 leaf stage of rice at the earliest and later on. So in the susceptible populations, we saw pretty reasonable control. Again, this is greenhouse screenings, but at least 70% for our granular applications at the beginning of the season, and between 40% for regiment up to 100% for propanil for the foliars. And similar results with the other susceptible population. Butte is not a really well-known grass herbicide. We did include this just because a lot of folks are using it at the beginning of the season, and I wanted to see what it looked like in the greenhouse. Okay, so for our, these are the samples that we collected that we were unsure of the sort of resistance status of these herbicides, whether or not they can control these types. So these 3 samples are from Sutter, and I realize they're not grouped together, but I just kind of wanted to give people an idea what it looked like. So you can see that our herbicides that we're putting out at the beginning of the season don't really control these particular biotypes, right? For the most part, the later cleanup herbicides still look pretty good. So this is not resistant. These are not resistant to these herbicides, the foliar herbicides. But yes, they are looking like we have resistance to our granular applications. And this really does align pretty well with what the growers are saying in the field. The folks that I've talked to, they're saying that when they put out their upfront herbicides, this grass seems to walk right through it. So this aligns pretty well with what we've been hearing in the field. Same thing with our other samples. So this is a susceptible compared to the Yolo County sample. And there's some variation on some of these samples. Some of the granulars are working, but for the most part, not working 100%. These are our Butte samples. So again, I'm giving you guys kind of an overview here. In some of the fields, Serrano appears to be still working fairly well, but for the most part, Granite, Bolero, and Butte really are not working as upfront applications. Glenn County, we had some variation. So, and I will address this in my last slide. So again, we're still having issues, I'd say, with applying, basically sort of how we are applying our herbicides, right? So there's still some folks that are misidentifying or thinking they have a herbicide resistance problem, when in fact it's most likely some sort of application error. So this one here on the left is likely an application error or some sort of issue with irrigation management, et cetera, et cetera. Okay, but the one on the right, again, we do have some resistance showing, right? We've got Granite, Bolero, and Butte really not working at all. So again, this is very preliminary data. There was only 10 samples. We collected a bunch of samples this summer, 64 additional samples that we are going to be looking at this coming year. But this, again, is some preliminary data that we can start to talk to folks about. So the early granular herbicides really do appear to be overall quite ineffective. I'm not going to say for certain that it's resistance in every case, but it might just be that this biotype naturally is not susceptible to these herbicides. The foliar applied herbicides appear to still have good control, propanil, regimen, and clincher. However, I did it at an early timing. So I did it at the 2-leaf stage of these grasses. So if you're applying later in the season, maybe not quite as effective. And that would be something that we need to probably test in the field. There's still some herbicide failures that are likely happening because of application or timing issues, like I said earlier. So we still need to work on our application methodologies. But it does appear overall that we do have some sort of resistance, or these are not susceptible to at least some of our herbicides. So like I said, we'll be doing a bigger screening this year. And also trying to gather some phenotypic information. So how do these plants look, and how do they look in comparison to what we know as sort of the species or biotypes that we've had in California for a long time. Okay, our weedy rice update. So I'm not going to spend a lot of time. This is the overall sort of map of all of our counties from 2016. Just to kind of remind folks where we are, we've got 7 biotypes. Actually, the 7th one isn't on the map, but 6 biotypes of weedy rice distributed pretty much across the entire rice growing region. There's close to 14,000 acres total infested, and we did do an update this year, which I'll talk about in a moment. Not sure what's going on here. I'm blocking. Can people see still? My screen says there's some sort of blocking happening. We did a field survey in 2019, like we normally do. by the end of the season. So this is an update from that other map. We did have a few new sites in 2019. One previous grower with 2 additional biotypes, and most of the new sites are type 1 weedy rice. And in 2019, we did have a new biotype, type 7. In 2020, we had 3 new sites, all type 1, one in Sutter, one in Yuba, and one in San Joaquin. We had previous growers also with new types. So that means they had at least one type already found at their site. And then this past year, they added another additional site. We also resurveyed all of our previously found acreage. I don't have the data for that yet, but we did that this year, and we'll be presenting that information to everyone later this spring. But it was actually a very good survey. We resurveyed all the fields that had weedy rice in the past 4 years to see sort of what the status was this year. And then we also surveyed fields around those fields to see if there was any weedy rice that had moved into the fields around them. I will give an update really quick on Suppress. So I've been talking about Suppress, which is a herbicide, an already registered herbicide in California from Westbridge. In 2019, we did some field testing on a type 3 field. The efficacy was okay in 2019, and we believe that perhaps we sprayed a little bit too close to heading. In 2020, we did some additional testing in the field again with multiple weedy rice types. Efficacy appears good. It was applied before heading, so a little bit earlier than we did the application in 2020 or 2019. This year, we also did some greenhouse testing at 2 different timings, and this was all of the different weedy rice types in the greenhouse. And it did actually look pretty good in the greenhouse. So efficacy was 100% in the greenhouse on the biotypes that we tested, which were biotypes 1, 2, 3, and 5. And we did earlier and a later testing to kind of simulate what folks might be doing in the field, and it looked quite good. So the data will be passed to Westbridge, and I'll just remind everybody that at this point in time, the label does not allow for application when there's standing water on the field. So this is looking like a very good option for us, but again, it has to be applied when the field is drained. Again, maybe that will be changed by next year. I'm not sure. We'll have to see if the label, if we can get a label amendment. I'm not positive about that, but at least we know that it does work. But again, with that caveat that the field has to be drained. So just to take a look at what it looked like. So in 2019, we did see pretty good efficacy, but the plants did go on to recover, and there were some viable seeds in the seed heads, in the panicles. So again, the timing perhaps was too late, so we'll have to do an earlier timing if we want to use this in the field. This was 2020. This was a week after spring, and the application timing, this timing really looked pretty good. The plants we sprayed before they'd started to head, and by the end of the season, none of the plants had survived. They completely died, and none put out heads. So I think this timing is going to be the one that we probably go for. So it's when you can see the plants in the field, but they haven't begun to head. The other large experiment that we've been working on with weedy rice is looking at overwintering, and this is to see how weedy rice survives over the winter based on a couple of different field conditions. So the treatments that we've been doing this in basins, basically, swimming pools, actually, little like kid pools, to simulate winter flooded conditions. So we buried seeds types 1, 2, 3, and 5, and we also buried M206 as a control, as a comparison. And then we did 2 depths, soil surface, and then 6 centimeters deep. The depth at 6 centimeters was supposed to simulate tillage. So sort of no-till in the fall if you left them on the soil surface versus 6 centimeters, which would simulate fall tillage. We did a ambient weather, which is basically just leaving the swimming pool to see what what would be happening with just sort of normal winter conditions, and then flooded conditions. They were removed from the soil at 30, 60, and 90 days after burial. And then we evaluated them for germination rate, dormancy, and viability or death after we removed them from the soil. Okay, I'm not going to go over all the different types, but I'm just kind of looking at what is happening over the 90 days that they were in the soil. So 30, 60, and 90 days after burial, this was the average percent of dormant seeds. And I kind of just wanted to go over this type or this particular evaluation real quick. So basically, overall, when seeds are buried under the ground, there's dormancy. So meaning that if you reflooded your field in the spring, if you had tilled in the fall and you flooded your field in the spring, most of the seeds wouldn't come up, okay, if you had done tillage. So in this case with type 1, we're inducing dormancy to about 16% of the seeds. So about 16% of the seeds wouldn't germinate. And this, you might be asking why this is bad. Basically, it's bad because it's going to prolong the length of time that those seeds are in the field. So if the more of the seeds are dormant, they're going to be in that field for a much longer time. They're not going to germinate that first year. They might germinate 10 years from now when they're brought closer to the soil surface again. The other thing about this, though, is that burial also seems to encourage seed death. Okay, so we're seeing seed death with a little bit of tillage in the fall, but also increased dormancy. So we'll be repeating this year, and we'll kind of see how these two things interact a little bit more. But you can see 30, 60, and 90 days after burial, we're seeing an increase in seed death, okay, in our buried seeds. So increase over time to up to about 60%. So, and again, last year there wasn't a lot of rainfall, but there was some. This year is looking like it might be a little bit drier, so maybe we'll see some more differences between the two treatments. Okay, just to quickly go over, there are a lot of differences between the biotypes. So this slide is a bit busy, but just to kind of give folks an idea, there are some biotype differences. So I'll be, trying to summarize the data along with some of the data that we get this year, next fall, to give everyone a little bit more of a conclusive idea of what the differences are. We might have different recommendations for some of the types versus others. So for type 5, and this corresponds pretty well with what we're seeing in the field. So type 5, there is no dormancy. This particular type isn't a dormant weedy rice. So basically, any sort of fall tillage is actually going to be an improvement in control for type 5. So tilling in the fall for type 5 would be recommended because it doesn't have any dormancy. For the other types, tilling in the fall, you can kind of see this is the tillage in gray here. Tilling in the fall is actually going to induce more dormancy, and again may prolong the amount of time that you're having these seeds in the field. So for type 1 and type 2, fall tillage is probably not going to be a recommendation. These were the seeds. The previous screen was the seeds that were in the flooded conditions. These are seeds that are in actually controlled or ambient conditions. And I think the biggest difference here is with type 5. So we've been recommending flooding up to this point in time. And again, this is preliminary data. This is just 1 year's worth of data. But it's looking like with type 5, actually burial and no flooding of the field is going to kill the largest percentage of seeds. So this in here in yellow is the number of seeds that died over the winter. This was in a non-flooded fall-tilled field. So this is 81% of the seeds actually died under these conditions. So in that case, doing that would actually be probably one of the quickest ways to get this type of weedy rice out of your field. So again, there's quite a lot of differences between the biotypes. And again, this is preliminary data, but I think it's going to give us some interesting, ideas of how to deal with weedy rice over the winter, especially if someone has a bad infestation. This might actually be a really great way to deal with weedy rice. Okay, so for preliminary results of this, again, we're repeating this a second time. So generally, length of time that the seeds are in the field, longer time equals more seed death. So over, making sure that you're harvesting and doing all of your field prep pretty much as early as you can in the fall in those weedy rice fields. And then if you can delay planting in those fields in the spring, that might also help to kill a few more seeds. Again, there's some significant differences between our biotypes, and more specific data on that will be coming next year. And then the last important point is that burial at 6 centimeters induces dormancy in some of our types. So basically type 1, type 2, type 3, dormancy is induced by any sort of fall tillage. So possibly lengthening time in the field. But there is a trade-off, and again, we'll have to kind of tease out how these two things interact. It appears also to increase seed mortality or seed death. So some interesting data, I think, and more to come next year. And I think that's the end for me. So I've got a few minutes for questions, and we'll also do the poll.
Okay, Whitney, you had one question in the chat. Oh, I'll let you do the poll first.
Okay. Maybe I can, or do you want me to answer it while the poll's running, or?
Sure, if you want to, you can. I can. Michael Horak asked, how are the infested acres estimated? Is the whole field counted or just the part of the field where the weed is present?
All right, good question. So actually, that was part of the reason we did this survey, like bigger survey this year. So in the past, we've been counting acres as basically if we found even one weedy rice plant in the field, or I think we were doing it by check before. So if we found one weedy rice plant in a check, that entire check would be counted on the map as acreage. For this survey that we did over the summer this past year, we actually made a scale, and Luis, you can jump in if you want to add anything here, but we made a rating scale basically if the field had one plant versus 60% of the field being infested, those would be accounted for on the map. And we did it by check this year as well, so that each individual check in the field was surveyed separately for infestation level. So the data that we got this past year will be much improved versus the data that we have from the past 4 years. So hopefully that answered the question.
Okay, and you have another question. What rate and water volume of suppress?
Oh, okay. So I actually went with the highest labeled rate on the label, and I can't remember the exact labeled rate, but whatever was the highest labeled rate allowed. For water volume, I did 20 gallons per acre, and it was done with a backpack. So, a backpack volume.
Okay, that's all the questions I see for now, but feel free to type your questions into the chat, and Whitney can answer as the webinar progresses as well. And be sure to fill out the poll.
Okay, it looks like most people have answered, but go ahead. Sorry, Sarah.
Nope, go ahead.
Oh, I was just going to say, it looks like most folks have answered. If everyone who hasn't answered wants to real quick give an answer, Remember, this part doesn't count. Doesn't matter if you get it wrong or right, but it counts that you participated in the poll for your CE credits. Okay, we'll give everybody maybe 10 seconds more and then we'll end the poll. Okay. All right, so it looks like folks did pretty well. So preliminary data about the new watergrass species biotype suggest that it's both A and B. So it typically isn't controlled by early into the water granular herbicides. And it typically, when propanol and clincher are applied early, they appear to have efficacy. And then question number 2, fall tillage induces dormancy of weedy rice. That is true. And that's it. So good job, folks. And again, those questions will be shown in your your test that you have to take after this. Okay.